Heterochromatin protein 1, also called HP1, recognizes H3K9 methylation and induces chromatin condensation through the recruitment of other chromatin remodelers. In mammalian cells, HP1 has 3 homologs, HP1 alpha, beta, and gamma. They share the similar structure of chromodomain and chromoshadow domain, but the length of hinge region linking these two domains are different from each other. As we can infer from its name, HP1 plays a key role in heterochromatin formation. But recently, it has been reported that HP1 gamma can be localized at the euchromatic gene region. So in this study, I have tried to reveal the gene regulatory role of HP1 gamma during cancer progression. So firstly, we analyzed the expression patterns of HP1 alpha, beta, and gamma in diverse cancer tissues using the public data in Human Protein Atlas. And we found that HP1 gamma was abnormally localized in the cytoplasmic region in many cases of cervical cancer. As you know, cervical cancer is caused by the recurrent infection with human papillomavirus. Among many subtypes of HPV, high-risk HPV, including HPV 16 and 18, is considered as a major cause of cervical cancer because they express the oncogenic protein E6 and E7, which inhibit the actions of tumor suppressors like p53 and RB, respectively. So we evaluated the expression pattern of HP1 gamma in cervical tissues. Interestingly, the HP1 gamma was diffused to the cytoplasmic region in cervical cancer tissues, while detected only in nucleus in normal cervical tissues. Nuclear fractionation assay also showed that the expression of oncogenic E6 protein increased the cytoplasmic diffusion of HP1 gamma. And knockdown of E6 protein in cervical cancer cells increased the nuclear accumulation of HP1 gamma. GEO profile analysis showed that exportin-1 expression was highly increased in the cervical cancer tissues and cervical cancer cells. Exportin-1 is a transporter that is responsible for the nuclear export of many proteins. So next, we tested whether HP1 gamma can interact with exportin-1. These data show that overexpression of HPV E6 induce the interactions between HP1 gamma and exportin-1. So next, we tested the effect of exportin-1 inhibitor, leptomycin B. Upon the treatment with leptomycin B, interactions between HP1 gamma and exportin-1 was decreased, inducing the nuclear accumulation of HP1 gamma. And leptomycin B treatment increased the p53-dependent apoptotic gene expression, which can induce the apoptosis of cervical cancer cells. So next, we identified the sequence of nuclear export signals within the HP1 gamma protein using the prediction solver. And we generated the mutant vector of HP1 gamma, in which the leucine-rich site recognized by the exportin-1 was substituted by the alanine mutation. This mutant failed to interact with exportin-1, and thus the vector was detected only in the nucleus. And expression of mutant vector increased the p53 signaling. And then we tested the tumorigenesis of HP1 gamma expressing cervical cancer cells. And these data show that overexpression of HP1 gamma decreased the tumor size, and NES mutant HP1 gamma was only detected in the nucleus, showing the more anti-tumor effect compared to wild type. And then to investigate the gene regulatory role of HP1 gamma in the nucleus, we performed microarray screening. And we found that UB123 is a candidate target gene of HP1 gamma. UB123 is a ubiquitin conjugating enzyme that is responsible for the HPV-mediated p53 polyubiquitination and degradation. Knockdown of HP1 gamma increased the UB12/3 expression and polyubiquitination of p53, decreasing the protein level of p53 and the expression of p53 target genes. And these effects of knockdown of HP1 gamma were all recovered by the co-knockdown of UB12/3. These data suggest that HP1 gamma can promote p53 signaling through the suppression of UB12/3 expression. In contrary to the knockdown of HP1 gamma, overexpression of HP1 gamma suppressed UB12/3 expression and p53 polyubiquitination, increasing the p53 signaling. Not only in HeLa cells, in other HPV-positive cervical cancer cells like SHIHA and KASKI, overexpression of HP1 gamma suppressed UB12/3 gene expression, increasing the p53-dependent apoptotic gene expression. But in HPV-negative cells, overexpression of HP1 gamma did not affect UB12/3 and p53. However, upon E6 overexpression in HPV-negative cells, expression of HP1 gamma suppressed UB12/3 and increased p53 signaling. Through this study, we identified that HPV infection induces the abnormal nuclear export of HP1 gamma through the exportin 1 and thus inhibit UB12/3 suppression and increases the excessive degradation of p53, which can induce cervical carcinogenesis. And we also found that NES mutant HP1 gamma successfully suppressed the UB12/3 gene expression and thus increased the p53-dependent apoptosis of cervical cancer cells. They are my colleagues, and especially I would appreciate Professor So Hee-kwon and Jun-jung Choi for helping me with this project. Thank you for listening.